this video, we will be going over the 2013 AP Biology free response questions 5 through 8. Question 5. The table below shows the amino acid sequence of the carboxyl terminal segment of a conserved polypeptide from 4 different but related species. Each amino acid is represented by a 3-letter abbreviation, and the amino acid residues in the polypeptide chains are numbered from the amino end to the carboxyl end. Empty cells indicate no amino acid is present. Part A. Assuming that species 1 is the ancestral species of the group, explain the most likely genetic change that produced the polypeptide in species 2 and the most likely genetic change that produced the polypeptide in species 3. So looking at species 2, we can see that the sequence is identical to that of species 1, except that on position 4, where there should be a valine, there is a lysine instead. And this is most likely due to a small-scale mutation, probably like a point mutation, a substitution. And then looking at species 3, we can see that the chain ends early. Its C-terminus is at the 8 position instead of at the 10 position like the others. And this suggests that there was a premature stop codon that was introduced into this polypeptide through a mutation. Most likely due to a deletion or frame shift or something like that. So the translation is terminated early. Part B. Predict the effects of the mutation on the structure and function of the resulting protein in species 4 and justify your prediction. So looking at species 4, we can see that up until residue number 4, everything looks good. But then starting from position 5, it all gets messed up. Like all these residues are not what they're supposed to be. And so this is an example of what's called a frame shift mutation, where when you add in an amino acid or delete one, the reading frame of the ribosome is completely different now. So all the amino acids after the site of that mutation are going to be completely altered. So this is probably going to cause a complete or large or partial loss of function in the protein because so many residues are being changed. It is likely that some of these are involved in the functional sites of the protein. But they said this is near the C-terminus, but it is likely that some of these residues will be involved, and the side chains of the amino acids mean everything in the function of a protein. If some of these amino acids are altered, that could completely change the protein's function. So no matter how small this altered segment is, it could still be important. Question 6. The following data were collected by observing subcellular structures of 3 different types of eukaryotic cells. Based on an analysis of the data, identify a likely primary factor of each cell type and explain how the data support the identification. So looking at cell X, we can see that it doesn't have a lot of smooth ER, it doesn't have a lot of rough ER. It has a large number of mitochondria, it has cilia, and it has few Golgi bodies. And so what are the organelles that are not present in very large amounts? That would be the ERs and the Golgi bodies. And those organelles are involved with the secretory pathway, so we can assume that this type of cell does not do very much secretion or processing of proteins. Instead, it has a large number of mitochondria, which provide energy to the cell, and it has a large number of cilia, which provide locomotion. So we can assume that this cell, most likely, its purpose is to convert chemical energy from the food that it takes in to mechanical energy with the cilia, to most likely for locomotion, or like the cells in the trachea that have beating cilia as well. Next, let's look at cell type Y. So it has a large number of smooth ER, a large number of rough ER, moderate mitochondria, cilia are absent, and a large amount of Golgi bodies. So all the organelles, like most of these organelles that are involved with the secretory pathway, are present in large amounts. So it's likely that cell Y is involved in secretion or processing because the large number of organelles associated with the secretory pathway that are present in a large amount. And finally, let's look at cell Z. So as you can see, pretty much all these organelles are absent in cell Z. So it's unlikely that it's going to be doing very much in terms of like processing proteins, producing proteins, or locomotion, or like reproduction or any of that. So it's just it probably just has a nucleus or no nucleus at all. It's so it could be for purely structural functions. There are a lot of cells like this in plants that are just there for structural purposes. They don't actually do anything in terms of like transport and that kind of stuff or signaling. They're just there to support the organism. Part 7. In an experiment, rats averaging 300 grams of body mass were tested several times over a 3-month period. For each individual rat, urine was collected over a 3-hour period after ingestion of 10 milliliters of liquid, whether it be water, 1% ethyl alcohol solution, or 5% ethyl alcohol solution. The volume of urine was then measured, and the results were averaged for all individuals within each experimental group. The data are shown in the table below. Part 8. Pose one scientific question that the researchers were most likely investigating with the experiments. So let's look at the variables that we have in this experiment. So our independent variable, the thing that we are changing, seems to be the concentration of alcohol in the fluid that was ingested. And the average urine output is our dependent variable, what we're trying to measure. So it's reasonable to assume that these scientists are trying to see if differences in alcohol concentration affect the level of urine in these rats, or and this also, or it could also mean that they're trying to look at the function of rat kidneys. And another thing, because researchers usually use rats and other animals in place of human subjects, they might just be trying to make generalizations about mammals as a whole, just using these results from the rats. Part B. State a hypothesis that could be tested to address the question you posted in Part A. So in Part A, we said that the scientists are trying to say what effect, if any, does increasing the alcohol concentration have on the urine output. So you could literally make any hypothesis as long as it involves a cause and effect. So you can say that either an increased/decreased alcohol consumption will either increase or decrease, or it does not affect the urine output in rats. It doesn't matter what you write here, because they just want you to make a hypothesis. They don't want you to make something that is likely to be true. They just want a hypothesis. But then part C is where you really have to reason with the data you have. So you can see that as the concentration of alcohol increases, the average urine output increases. So we can reason that the increased ethyl alcohol consumption increases the urine output in rats, and likely in all or most mammals. Finally, question 8. The figure above represents a generalized hormone signaling pathway. Briefly explain the role of each numbered step in regulating target gene expression. So in step 1, we have what seems to be a molecule that is binding to a transmembrane protein. Now, if this transmembrane protein has a molecule binding to it, it's most likely a receptor, because the description says this is a signaling pathway. And signaling pathways usually begin, if it's a hormone signaling pathway, usually begins with the hormone activating a receptor on the membrane of the cell. Then in step 2, we can see these arrows going to many different molecules. It's like a chain, and going all the way to the nucleus, and this is going to be where the signal is passed down. It's called a signaling cascade. And so this signal is going to be passed down between all these different molecules, and it might be amplified throughout the cell as well. And it's going to go all the way down to the nucleus, where it's going to alter the expression of what seems to be either an activator or a repressor protein. It doesn't matter which for this question. And this is going to affect the gene expression in the nucleus. Thanks for watching this video, and please subscribe for more AP Biology content.